Okay, so we just got done talking about excitation-contraction coupling, and that is the link between the excitation, which is the action potentials. How do we generate action potentials on the sarcolemma, right, along the plasma membrane of the skeletal muscle cell? How do we get from that electrochemical message to actual contraction? And the link is the release of the calcium, right? So in order for muscle contraction to happen, we have to get that calcium released. And so we left off on the last video with, okay, an action potential gets generated along the length of the sarcolemma, and that action potential, that wave of depolarization, will travel down not only the entire length of the sarcolemma, but also down those tubule invaginations of the sarcolemma called the T-tubules or the transverse tubules. Sandwiched on either side, on both sides of those T-tubules, we have those enlarged regions called the terminal cisternae of the sarcoplasmic reticulum, and the sarcoplasmic reticulum stores calcium. And remember that action potential causes voltage-gated, when it arrives at the T-tubules, it causes voltage-gated calcium channels on the SR to open. We get a release of calcium ions from the sarcoplasmic reticulum, right? And that calcium, those calcium ions are going to bind to troponin. So if you remember me talking about in the previous, very beginning videos of chapter 10, we talked about thick and thin filament structure. The thin filaments, the actin filaments, look like beads on a string. So these are individual globular actin subunits that bind together and form this long filamentous protein, which are two strands wound together, right? And then there are regulatory proteins attached to the actin. So the purple is the actin, again, globular actin subunits that are arranged like beads on a string, and there are two strings wound together in like a helical structure. The regulatory proteins are the blue and the yellow. So tropomyosin is the filamentous proteins that cover the active sites on actin. So the actin active sites are covered by tropomyosin. That's a regulatory protein. This small globular protein that's made up of three subunits, this is a regulatory protein called troponin. And if you remember from those previous videos, I said that troponin has three binding sites, or three subunits, rather. And they each bind something different. So there is a tropomyosin binding subunit. It's going to bind tropomyosin. There's another subunit that will bind calcium, and then a third subunit that will bind actin. And that is what is stated here. So troponin has three subunits. One subunit binds calcium, one subunit binds actin, the other subunit binds tropomyosin. When calcium gets released from the terminal cisternae, it can bind to troponin on that subunit. So calcium binds to troponin, and it causes a change in shape. And when troponin changes shape, because it's bound to actin and it's also bound to tropomyosin, calcium causes it to change its shape and it slides tropomyosin out of the way, but only when calcium is bound. So when calcium is bound to troponin, it changes its shape and it slides tropomyosin out of the way and exposes the active sites on actin. So if calcium is not bound, so in the absence of calcium, tropomyosin covers the actin active sites. But when calcium is bound to troponin, troponin changes shape and slides, so look over here, this is calcium being bound, it slides tropomyosin out of the way, and now these active actin sites are exposed. And what can bind to these actin active sites? The heads, those globular heads on myosin. So this is what this diagram is showing. Here, a wave of depolarization, that action potential arrives at the T-tubule. That causes the voltage-gated calcium channels on the the terminal cisternae to open. The SR, the sarcoplasmic reticulum, stores calcium, so there's a higher concentration of calcium in here than in the cytoplasm. So calcium rushes out. Where are these triads located? They're located all around myofibrils. So when calcium gets released, guess what? They're right next to the myofibrils made up of actin and myosin. So they're in very close proximity to actin and troponin. So when calcium gets released, then calcium will bind to troponin. Troponin will change shape. Because troponin is also bound to tropomyosin, when it changes shape, it slides tropomyosin out of the way and exposes the actin active sites. And now myosin can bind. So calcium is the link between excitation and contraction. Calcium must be bound to troponin in order for these active actin sites to be exposed and for myosin to bind to actin. And only when myosin can bind to actin can we get contraction. And so here is a video showing that.
After calcium ions are released from the sarcoplasmic reticulum during excitation-contraction coupling, they are available to help initiate contraction. To understand how muscle filaments prepare for contraction, let's review the structure of a thin filament. Recall that it consists of two intertwined strands made up of actin molecules, much like a necklace made of pearls. A regulatory protein called tropomyosin spirals around each actin strand, and a second, smaller protein called troponin holds the tropomyosin in place. In a resting muscle, tropomyosin covers the regions of actin known as the active sites. As a result, it is impossible for myosin from the thick filaments to bind to actin and engage in contraction.
So when calcium is absent, right? When there is no calcium, troponin is in a particular shape that causes tropomyosin to cover the active actin sites on actin.
When calcium ions become available, they bind to troponin and cause it to shift its position. Troponin in turn moves tropomyosin off actin, thus exposing the active sites. Actin is now available to bind to myosin, and the muscle can contract via a sequence of events called the cross-bridge cycle.
So calcium, these little green ions, bind to troponin, and that changes the shape of troponin, and it slides tropomyosin out of the way. And so now all of a sudden, the actin active sites are exposed, and myosin can bind. All right, so tropomyosin. All right, so which of the following is going to be true of tropomyosin? Tropomyosin covers actin active sites? Yes. Binds calcium? No, that's what troponin does, so that's incorrect. Is a small globular protein? Nope, that's incorrect. Remember, tropomyosin are those long, slender filamentous proteins that wrap around actin. Has 3 subunits? That's incorrect. Troponin has 3 subunits. Troponin has a calcium subunit, an actin subunit, and a tropomyosin subunit. So the correct one here is tropomyosin covers active actin sites. Troponin has 3 subunits. Okay, that's correct. Which of the following does not bind to one of these subunits? So remember, troponin has 3 subunits. Which of the following does not bind to one of these subunits? Actin, that's correct. Myosin, calcium, I know that's correct. Oh, tropomyosin, yes. So troponin is going to bind to actin, it's going to bind calcium, and it's going to bind tropomyosin. So myosin here is incorrect. It's tropomyosin that it binds, which is one of the regulatory proteins. So troponin's 3 subunits. There's a subunit that binds actin, there's a subunit that binds calcium, there's a subunit that binds tropomyosin. Choose the correct sequence of events that occur in preparation for contraction. I like asking questions like this. And sometimes I'll ask questions where I will give you, something that just happened, and you have to pick which would happen. I'll give you then choices A, B, C, D of what would happen immediately after the step I just gave you, and you have to choose that. So that's how I may ask it as well. These sequence of event type questions. All right, so choose the correct sequence of events that occurs in preparation for contraction. So let's just look at the first. This is how I like to approach these questions. I will just look at the first part, right, the first step of each, and see if there are any that I can just immediately get rid of. Action potential arrives at the triad. Well, that sounds like, okay, in preparation for contraction, I'm going to keep this as a possibility because, you have to have an action potential arriving at the triad in order for contraction to happen. So tropomyosin exposes the actin active sites. Well, I know that there are several steps that have to happen before tropomyosin exposes the active actin sites. Right? There's other steps before this. So most likely this is not correct because this is not going to be the first step. And if you look at the second step of this sequence, calcium binds to troponin. Well, that has to happen before tropomyosin exposes the active actin sites. So this is most likely incorrect. Calcium is released from the terminal cisternae. Well, that's one of the first things that has to happen in order for contraction, but here, A gives me action potential arrives at the triad. That actually happens before calcium is released. So most likely this is going to be the correct answer because this has to happen before the calcium is released from the terminal cisternae. Let's just look at D to make sure. I always like, even if I have an idea that this is correct, I like reading through all the options. Calcium binds to troponin. Well, I know calcium has to be released before calcium can bind to troponin, so this is incorrect. So I've eliminated B and D just by looking at the first and second step of these two. All right, calcium is released from the terminal cisternae. Action potential arrives at triad. This has to happen before this happens. So A is the correct answer. Action potential arrives at the triad. Remember that action potential causes those voltage-gated calcium channels on the SR to open. That means calcium gets released from the terminal cisternae into the cytoplasm. Calcium then can bind troponin. Troponin changes shape. It shifts tropomyosin out of the way and exposes the actin active sites. So that's how you can approach questions like this by process of elimination. So skeletal muscle contraction. Now we have calcium bound to troponin. The next video, I'm going to talk about how the actual contraction takes place while calcium is bound.